the goal of the Ready AViD Center is to develop oral broad-spectrum, direct-acting antivirals against high-priority viral pathogen families. And so a question that we get a lot around this approach is, why broad spectrum? And I want to define broad spectrum, because that's another thing I've learned over time, is that people have different definitions. So when we talk about broad spectrum, we're talking about a therapy, or in our case, small molecules that would work against multiple viruses in the same family, just to set some terms. When we talk about things that would work against multiple viruses from different virus families, we refer to those as pan-viral. I was asked the other day how we came to our definition. Basically, we made it up because we needed to have common terms within the program. So that's what I mean by broad spectrum. But why broad spectrum? And I think this is pretty straightforward. First of all, a broad spectrum drug that worked against all the viruses in a family would provide treatments for diseases caused by those viruses in that family. And as we all know, for most viruses of concern, human pathogens, there are no antiviral drugs or therapeutics available. But the other thing about a broad spectrum drug would be, if it works, imagine a drug that worked against all the viruses in a family right now. When the next virus emerges, there's a very high likelihood it would also work against that next virus. So instead of, if it works against SARS, MERS, PEDV, and SARS-CoV-2, there's a higher probability that it might work against SARS-CoV-3. But the trick is to find a drug target that would be suitable to provide broad protection or broad inhibition of virus replication. And so what I want to talk to you today about is how we've been going about identifying and validating targets for broad spectrum antiviral drugs. There's basically three pieces. The first is in silico, where we're looking for highly conserved and potentially druggable pockets in viral proteins. We're going back in and doing the molecular virology and virology work to determine the role of those pockets in virus replication. And then using that information where we're able to validate a novel pocket to use virtual screens and eventually physical screens as well, to find new small molecules that bind that site and inhibit the virus. So where we're applying this, I'm going to talk to you about where we're applying this against alphaviruses. A little background about alphaviruses for those of you who don't know. So these are viruses that really fall into two different clusters based on their disease indications. One group, the arthritogenic alphaviruses like chikungunya virus, myrovirus, Ross River virus, is really characterized by this debilitating polyarthralgia, which can become chronic, sometimes lasting months, sometimes lasting years. On the other side, we have the encephalitic alphaviruses like Venezuelan equine encephalitis virus, Eastern and Western equine encephalitis virus. Again, the most prominent feature of these diseases is the encephalitis. Oh, I'm sorry. The reason that we're so concerned about alphaviruses is that there have been multiple widespread outbreaks of alphaviruses since they were first discovered in the 1950s. These viruses are endemic in some cases. Viruses like EEE and Western can be found here in the United States. But then there are viruses like chikungunya virus, which until around 2013 were actually not found in the Western Hemisphere, and then exploded to cause over 1 million cases in just 1 year or 18 months upon their introduction into the Caribbean islands. So in addition to these viruses, we know that there are viruses in the alphavirus family that are poised to emerge and spread widely through their mosquito vectors. Viruses that currently don't cause widespread outbreaks, but have the capability, either in their current state or with just a couple of mutations, like myrovirus in South America, for example. So new threats are on the horizon in the alphavirus family. But here's the problem we're all trying to solve. There are no antivirals approved to treat or prevent disease caused by any alphavirus. So a broad spectrum alphavirus drug would be a major advantage for all of those currently circulating alphaviruses, as well as future threats that may emerge. And a little bit here about the replication cycle. I won't go too far into this, but I think it's relevant for what I'll talk about. So alphaviruses are positive sense single-stranded RNA viruses. That means that the incoming genome is an mRNA, in essence. It's translated immediately. The first things that get made are these non-structural proteins, which are cleaved up by a viral protease encoded by the NSP2 gene, which I'll talk about for the remainder of the talk, into the individual subunits. And that activity is in fact required for the formation of the replicase complex. So NSP2, like almost every viral protein, as we all know, is essential in a positive sense. RNA virus, at least, they're typically essential. And it's multifunctional. So NSP2 really has two domains: an amino terminal helicase domain, with a short linker separating it from a carboxy terminal protease domain. And you can see the MTase-like domain or methyltransferase-like domain. It's not actually a methyltransferase. It just kind of looks like one. And it's actually, part of the functional protease domain on the carboxy terminus. So we have programs running now around hit discovery, hit to lead optimization, and lead optimization for these targets, the protease and the helicase. But our question was whether we could actually find additional target sites in NSP2 for broad spectrum drugs. So this is the approach that we came up with to do this. So a lot of times when people think about broad spectrum, they think about conservation. And I see a lot of talks where people focus on conservation at the level of the linear amino acid chain. But as we all know, that's not sufficient necessarily to make a good target site for a small molecule or other therapeutic. Instead, we need to think about how a small molecule might engage with the tertiary structure of that protein. So we use AlphaFold to do structural modeling of all the different NSP2s from across the alphavirus family. And then in parallel, we use a program called FTMap, which basically looks at potential ligand binding hotspots on the surface of a protein. And of course, then we overlay those two pieces of information with conserved amino acids. What we end up with is a select number of sites on, in this case, the NSP2 protease domain that satisfy all these criteria. Structurally conserved amino acids conserved at a higher degree than over the rest of the protein, and the potential to be small molecule binding sites. So just to give you an example, one of the things that is sort of a prerequisite for our approach is a domain or protein where we can benchmark to know that our in silico predictions of structure in fact match what has been observed. So using chikungunya virus protease domain as our benchmark, we found very good similarity to the actual X-ray crystal structure that's been determined. And we found that based on that, most of the other alphavirus protease domains that we produce structures for using AlphaFold 2 showed a very high degree of similarity, despite, as you can see on the right, a fair degree of amino acid differences across the protease domain. So of course, just finding these sites is interesting, but not sufficient. So I wanted to talk about our steps going forward to validate these sites. And actually, the mutagenesis strategy is worth note. So when we went into these conserved pockets, we then went and looked at the underlying residues and specifically designed point mutations that are predicted, and I have to emphasize predicted until proven otherwise, to alter the pocket without disrupting the overall structure of the domain or protein itself. So once we have those mutations in hand, together with the computational analysis, we can actually go in and start to see what happens. So here we're using an infectious clone of chikungunya virus. We're introducing the mutations I showed you on the previous slide into the NSP2 protease domain in the context of the viral genome. And then we're doing two assays. The first is a specific infectivity assay, which basically measures replication over one round, where we electroporate in vitro transcribed RNA into cells, and then we see how many of those cells give rise to plaque-forming units. And in parallel, we take some of those cells and we let them grow for a while and see how much virus comes out. So you can see there's a wide range of outcomes. One takeaway already at this point was that just because you're conserved, you look like you can bind a small molecule and you're structurally conserved, doesn't necessarily mean, at least in this cell type and in these assays, that you're required for virus replication. But we did see a few areas where we saw fairly dramatic impacts on both specific infectivity and on viral titer. So going forward, to kind of zoom in onto one of these pockets that a graduate student in my lab and Mark Heisey's lab, Jack Sears, really focused on, the brown/red pockets, so they're adjacent to one another. And as you can see in the inset, what Jack noticed when he looked at the structures is that there appeared to be hydrogen bonding across 3 different residues that he hypothesized might be important for function. So he went in and made a series of mutations that disrupted these amino acids or change them. And in fact, he did see an impact on specific infectivity and titer. He was also able to, very interestingly, generate temperature-sensitive mutations. So these are mutations that allowed the virus to grow at a lower temperature, 30 degrees, but not at the higher or normal temperature of 37 degrees. I view that as a suggestion that there may be some change in the structures, but of course, why that TS mutant is in fact TS remains to be determined. And again, as I showed you before, these more refined mutations also showed impacts on virus yield. So this was very interesting to us in that it appeared that we had found a new domain that could potentially bind a small molecule on the surface of the chikungunya virus NSP2 protease domain. But remember, our goal was actually to think about broad spectrum. So we need to know more information, not just the impacts of mutating this site on chikungunya virus, but on more distantly related viruses. And the ones that we looked at further were Ross River virus, Symbis virus, Venezuelan equine encephalitis virus, and Eastern equine encephalitis virus. We did not take on the salmon pancreas disease virus just yet. We thought it might be out of scope for AViD. So as you can see, when we made analogous changes, so you notice the numbers are a little bit different. That's because, remember, these are structural information went into these predictions. When we made the analogous changes in Ross River virus, we again saw a defect in both specific infectivity and titer. Interestingly, that defect in titer was less pronounced for Venezuelan equine encephalitis virus, but was still, in fact, fairly significant, particularly here at these boundary amino acids for V, and similarly for Symbis. And also when we went forward using an Eastern equine encephalitis virus replicon system, we again saw significant impacts on yield when we've made these mutations, again, focusing around this Y727 and these amino acids thought to drive the hydrogen bonding interactions. I will note, I didn't think I'll have time, and I won't to present today. We have gone back, and in cases such as in V, or where we see large differences in titer, we've gone back and sequenced those viruses, and what we've in fact found are reversions, either back to the wild type original amino acid, or in some cases to very similar amino acids, which suggests that there is a real pressure on these viruses to maintain those hydrogen bonding interactions. We then went back and asked, how do these mutations impact known functions of NSP2? And the two I mentioned at the beginning were ATP-dependent helicase and then a protease activity. Remember, the ATPase domain is that amino terminal, so separate from the piece we've been looking at. What we found, not surprisingly, is that the mutations that we had made to these pockets in the protease domain were not impacting the helicase activity or the NTPase activity of NSP2. But in fact, when we went back in and looked at their impact on protease activity, we saw a wide range of effects, suggesting that some of these novel conserved pockets, and I'd call your attention again to this Y727, tyrosine 727, where this mutation has almost completely ablated protease activity, even though it is not within the active site of NSP2. So this could suggest that some of these other pockets in the protease domain that are highly conserved and potentially sites for small molecule binding are regulating the activity of the protease through a mechanism other than directly impacting catalytic activity. However, caveat, when we do experiments like this, we put in amino acid changes into a protein that is essential for replication. We don't even know at this point in time if this protein is folding correctly. So the last thing Jack went back and did was ask that question. And we actually ran 3 different assays together with help from the folks at Penn State University's core, looking at the effects of the one mutation, the tyrosine 727F mutation, on overall structure using differential scanning calorimetry, analytical ultracentrifugation, and SAX analysis. And what we found from that is the data is consistent with no gross or overall change in the structure of the NSP2 protease domain, as we had predicted based upon our mutagenesis strategy. So this is going to be brief because there's not a lot here yet. But what we did then is we took that information and we specifically masked off the rest of the NSP2 protease domain. And a protease was, a virtual screen was run on this domain. We were able to find small molecules that were predicted to interact. Oh, Jack, put in an animation on me. But when we went back and tested for the effect of those small molecules on either protease activity, the ATPase activity, or in a live virus assay that expressed a nanoluciferase gene, as you can see, not a whole lot of impact. So, but this is, again, just a small number of compounds. I will point out, though, that we did find one small molecule that showed very weak inhibitions at high concentrations. All the caveats here, we would have predicted this would impact protease activity. However, we don't actually know if this small molecule is directly interacting with the protease domain. If so, where it's interacting with the protease domain. All we do know is that the screen yielded something with weak inhibition of virus replication. So a lot more work to be done here. But let's just say Jack was excited that 1 out of 43 compounds at least showed promise. We are applying this approach more broadly across our program as well. So this is another Alphavirus target. This is the NSP3 protein. For those of you who think about alphaviruses a lot, this is the protein that contains the macrodomain or the ADP ribosyl hydrolase. There's a lot of efforts going on that target, the macrodomain. We actually focused our analysis of target sites on a carboxy terminal piece of the protein called the AUD or alphavirus unique domain. Taking the same basic approach, looking at structural conservation, looking at amino acid conservation, and then using FTMap to identify potential small molecule binding sites. And again, as you can see, we were able, using the specific infectivity assays and the replication assays, to identify point mutations that significantly impacted virus replication. I think the piece on the bottom, not my story. So again, I would highly recommend, if you're interested in this approach and learning more about it, either reaching out to Vic Say at St. Louis University or maybe having him come talk here. Because there he's had they've taken it to the next level, where in addition to using the same approach to find a novel conserved pocket on the surface of the dengue RNA-dependent RNA polymerase, the virtual screening has been more successful. They've actually found a binder that interacts not with the catalytic side of the RDRP and does impact dengue virus replication as well. So with that, I'll just kind of sum up what I talked about today about our approach that we're using to identify novel potential broad spectrum inhibitor sites, starting with our in silico analysis, moving forward and using specific mutagenesis strategies to test the role of these sites in the replication of not one, but many alphaviruses. From there, looking at impacts on known activities and structures, and then transitioning that towards virtual screening to find new molecules. Number 6 is a teaser. This will go back to the beginning, not the medicinal chemist. So since I figured there would be a lot of MedChem questions, I'm pushing this off to someone from our MedChem core to deal with. But we have some really exciting small molecules that are showing broad activity coming out of a traditional high throughput screening campaign focused on the NSP2 protease as well, and some novel results also coming out of some DEL studies that have been done. So for all of us in AViDS, we know one thing. Even a 20-minute talk has a big cast of characters that made it happen. Right there in the middle, in the biggest name is Jack Sears. He's in my lab and the Heisey lab. He made this. I don't know why he put himself in my lab. Normally they claim being in Mark's lab over being in my lab. He's done a tremendous amount of work. He's had a huge amount of help. I would definitely like to call out a few other people. Eric Merton at the Center for Integrative Chemical Biology and Drug Discovery here at UNC in Ken Pierce's lab, another graduate student who's done a tremendous amount of work testing small molecules. Konstantin Popov in Alex Tropsch's lab here did a lot of the computational modeling work together with Jack and his help with the virtual screening. And of course, Jamie Arnold and Craig Cameron, who were absolutely instrumental in setting up the assays that we're using to study the effects of small molecules on NSP2 function.